Hormone is abscisic acid. It is purely plant growth inhibitor hormone and antagonistic to most of the growth hormone, that is auxin, cytokinin, gibberellin. These are growth promoter, and to them it acts as in opposite direction. Means they are the plant growth promoter, auxin, cytokinin, gibberellin, and this hormone is having opposite effect to them, being it is inhibitory. The hormone which will work together to perform the same function, they are called as synergistic hormone, and the hormone which will work opposite to other hormones, so that is called as antagonistic. Like ABA is antagonistic to these auxin, cytokinin, and gibberellin. So if we see this abscisic acid, as I told you, it is inhibitor, and it is also called as dormine or abscisic 2. So these are the names. So during 1960, there was the two scientists that Adicott is there, and if we see the another scientist was that Warning. Two scientists was there, Adicott and Warning. So they reported three kinds of inhibitors, that is inhibitor B, then abscission 2, and dormine. These three chemical, and these were chemically identified as identical and now known as abscisic acid. So these three chemical. If we see chemically, this abscisic acid is dextrorotatory and it is cis Q terpene. This is dextrorotatory. Dextro means right, rotatory means rotation. You can see in the optical activity PPL. And if it is to rotate to left, that is called as levorotatory. Chemically, it is cis Q terpene. Q terpenes, this one. Or if we see about the abscisic acid, it is going to form from the violocyanthine, or we can say carotenoids, that because it's carotenoids, or formed from the mevalonic acid and xanthophyll. It is formed from the mevalonic acid and xanthophyll. Xantho it means yellow and phyll it means leaves. That we see in the four pigment, that is chlorophyll A, B, xanthophyll and carotene. So xanthophyll formula C40H56O. So this one is uh, from that xanthophyll this is going to form. So derivatives is carotenoids. Carotenoids we know carotene and xanthophyll. Then regulate the abscission and dormancy. These are its function. Then this hormone is also called as stress hormones. The another name for this ABA is also used, stress hormone. So it acts as inhibitor of the plant growth and plant growth metabolism. Then also inhibit the seed germination. It will make the seed to withstand for a particular time by promoting the dormancy. What is meant by dormancy? Dormancy means when the viable seed, living seed, fails to germinate in a favorable season. Even the season is favorable, but it is not going to germinate. The reason is being it is having the seed coat. So that seed coat is going to break by the two things, by naturally or by artificial means also. Then it will stimulate the closure of stomata. When closure of stomata is going to happen? In a xerophytic plant when the water is less. So to reduce the rate of transpiration in that plant, the stomata will remain closed during day and opens at the night. In that plant, CM cycle is going to happen, that is crystallization, acid metabolism. Xerophytic means the plant which grows in the dry desert area, arid area. So it will increase the tolerance, that it will stimulate the closure of stomata in the epidermis, then increases the tolerance of plant to various kind of stress. Hence the name is stress hormone. Then it has an important role in the seed development, maturation, dormancy. So seed dormancy in the ABA help to withstand desiccation. Desiccation means by water the loss is not going to happen to that particular part. Desiccation means if because of water if a particular part is going to damage, that is called as desiccation. So this desiccation is prevented by this ABA, and this seed coat will remain for a particular time, means dormancy will be continued with the help of this ABA. So that's why it is said that it will increase the, it will promote the dormancy and other factors unfavorable for the growth. में हम देख रहे हैं कि एब्सिसिक एसिड, तो क्या क्या फंक्शन है अभी मैं बोल चुका आपको कि जो एब्सिसिक एसिड है यहाँ पर तो इन कॉन्ट्रास्ट इनके अपोजिट ये काम करता है. अब्सिसिक एसिड इज एंटागोनिस्टिक टू दिस हार्मोन. ये बोल चुका मैं, ऑक्सिन, जिब्रेलिन, साइटोकाइनिन. ये कौन है? दीस हार्मोन्स आर ग्रोथ प्रोमोटर. और ये जो अब्सिसिक एसिड है ये ग्रोथ इनहिबिटर है. तो दोनों के फंक्शन कैसे हो गए? अपोजिट टू ईच अदर. एंड व्हेन बोथ द हार्मोन्स आर गोइंग टू वर्क अपोजिट टू ईच अदर, दिस इज कॉल्ड एज एंटागोनिस्टिक हार्मोन. एंटागोनिस्टिक. Antagonistic hormone means having opposite action. And these three hormones, auxin, gibberellin, and cytokinin, they work together. Hence, they are called as synergistic hormone. Synergistic means having the same kind of action, which stimulate the growth of abscisic acid. And then these are inhibitor also. I told you about the ABA, that ABA is also called as stress hormone, or the chemical found in it is abscission 2, that dormin 2. These are there, dormin. It was discovered by the scientist Adicott and Warning. Two scientists are there, very, and they named it abscission 2 or dormin 2 respectively. These names were given, but I told you there are the different names are also there. Then abscisic acid from the mosses and higher plant it was found, so on. Then chemically, if we see, it is dextrorotatory cis sesquiterpene. Dextro means right, and it is found from the, I told you, abscisic acid is going to form from the violocyanthil or mevalonic acid and xanthophyll, or their precursor is carotenoids. So this is major inhibitor. Remember this word, major inhibitor of growth in the plants, and it is antagonistic to the all the three growth promoters, especially the gibberellic acid. Because why it is said so? Because gibberellic acid and oxygen will break the seed dormancy, and this ABA will make the dormancy. It will promote the dormancy, and to break the dormancy, what is going to use? This gibberellic acid and oxygen and cold treatment, gibberellic acid and ethylene to break the seed dormancy. Then especially, then its precursor is violocyanthine, that I told you, xanthophyll in the chloroplast. Then abscisic acid is produced mainly in the mature leaves. This is where the senescence is going to happen. Then beside this, it is synthesized in the stem, fruit, seed, also transport to the rest of the plant through the vascular tissue. And some of its important role, now we are going to discuss it, some of the important role. So if we see the biopsy of ABA, biopsy means where it is going to found and how it is extracted. So that is about biopsy. Like during the cancer, that biopsy is going to happen like that. Then cotton or bean explant test. Then function. What is the function of ABA? So it Haston means, I told you. Haston means what? It will increase or speed up. It will hasten the formation of abscission layer. See, this is the stem, and suppose this is the branch on a plant, particular branch is there. Now, this part, being it is mature, and suppose this plant don't want this part, so it will make here abscission layer. And once the abscission layer is made, so this part is now going to, it is removed from the plant, and this is called as abscission. Now this part is going to remove. So this is abscission. This is called as abscission. So this is the function of this abscisic acid. What it will do? It will help in the abscission. Then it will promote the bud dormancy during winter. Bud dormancy also it will promote. Then transpiration, I told you, it will close the stomata during that hot climatic condition, means in a 0 condition, arid desert. So stomata will remain closed during day to prevent the loss of water by transpiration. So for that it is going to help. Stomata by causing the potassium ions, because potassium ions, carbonate, these are necessary for this kind of phenomena in the opening and closing of stomata. Leaves the guard cells and reducing their turgidity during the period of water shortage or drought, during drought condition, sukha, and hence known as stress hormone. Then it will promote the bud dormancy during the winter, that I told you. Then seed dormancy will induce the seed dormancy, hence named as dormine. Why it is called as dormine? So because it will induce the seed dormancy. And dormancy, I told you, what is my dormancy? If a viable seed fails to germinate during favorable condition, that is called as seed dormancy. And if the seed is going to fail, if the viable seed is going to fail during unfavorable condition, that is called as quiescent stage or quiescence. Quiescence. I discussed this about seed germination, everything in the previous video, so you can check out. Then help the seed to withstand the desiccation. Desiccation means loss by means of water. So that is going to prevented by this ABA. It will promote the seed dormancy. So seed coat will remain for a long time. That water is not going to break that seed coat easily, so that germination will happen and dormancy is going to break. So to continue the dormancy, so for that it is going to help. Then flowering. So in some short day plants which need the less sunlight, like strawberries. So in that plant, the flowering is going to happen with the help of ABA. Then again, two more functions are there, that is play important role in the seed development, maturation and dormancy, that we already discussed, and induce the synthesis of carotenoids in green oranges, making them yellow. See, we can see that raw mango is converted into a mango by solubilization of pectin, and that raw mango is green in color and mature mango is yellow in color. Raw lemon is green in color and when it matures it is yellow in color. So this color changes, this pigmentation is going to happen because of carotenoids. We know carotenoids means carotene and xanthophyll. Carotene is orange red color pigment and xanthophyll is yellow and phyll it means leaves. Remember this pigmentation is that what we say, album is white, colorless. Then chlorum is green, the xanthum is yellow, cyanum is blue, pheum is brown, and then chlorum is green. So these are the different pigmentation. Cyanum is blue, then orange, so on, different kind of pigment. So we saw the function of this